Good afternoon. Good afternoon, evening, wherever you might be. We're excited for today's webinar, your samples, your impact, and it's all about the Cure HHT Biobank. We're going to start by introducing our speakers. And first up, we have Dr. Cassie Friday. She's our Senior Director of Research here at Cure HHT. She leads our organization's research strategy to accelerate the development of new treatments for HHT, one of the most common inherited bleeding disorders. She works at the intersection of science, medicine, and patient advocacy, collaborating with research, industry partners, and people living with HHT to advance promising therapies from early discovery through clinical trials and ultimately towards regulatory approval. Dr. Friday earned her PhD in physiology from the University of Kentucky, where her research focused on Alzheimer's disease. After two of her children were diagnosed with HHT, she redirected her scientific career to rare disease research. Today, she is dedicated to building the infrastructure, collaborations, and clinical research programs needed to bring better treatments and a cure to the HHT community. Our second presenter is Cassidy Swords. She's our research coordinator here at Cure HHT, where she works closely with the HHT community to advocate for individuals and families affected by HHT and to connect patients with meaningful research opportunities that move us closer to a cure. Before beginning her career, Cassidy worked directly with individuals living with dementia, an experience that helped shape her passion for patient-centered care and advocacy. She has nearly 5 years of experience as a research coordinator, with much of her background focused on women's health research. Cassidy joined Cure HHT a year ago and is passionate about helping bridge the gap between patients and researchers, ensuring that the voices and experiences of the HHT community remain at the heart of research efforts. I'm going to pass it over to you, Cassie, and let you take it away.
All right, thank you. All right, let me share my screen here and get our slides up and going. Thank you so much, Phyllis. I am really excited to talk to you all about the Cure HHT Biobank today. We have a lot of different things to cover. So we'll talk about what a biobank actually is, why we need one for HHT research, what kinds of tissue we collect and store in the biobank, what that process looks like, kind of from your first interest in donating a tissue to sending it to the biobank and the post-processing of it. And then we'll also cover end-of-life donations. So a lot of people are interested in donating their body to medical research or to science, and we can do that in a way that's directed straight to HHT research. And then we'll talk a little bit about what the samples are actually used for, review how the biobank's actually doing today, and learn about some of the research that has come from human samples from patients with HHT so far, and then also go over the ways that you can donate in any kind of form. Donate tissue samples or help facilitate a donation of one of your patients if you are a healthcare provider who's listening. So first of all, the biobank is the place where we collect, store, and distribute biological specimens so that scientists can study HHT. The biobank is physically located in Boston, Massachusetts, in the laboratory of Dr. Andy List, who is an assistant professor of surgery at Harvard Medical School. And Dr. List studies pancreatic cancer, and he operates a very large pancreatic tumor bank at Massachusetts General Hospital. He has samples from over 2,600 patients in his biobank and a whole team of experts in pathology, tissue processing, and generating cells and all kinds of samples out of the pancreatic tissues from patients that come into his lab. So he's actually not an HHT researcher, but he's generous enough to rent some of the space in his lab and use some of his expertise to help rare disease organizations like us and help us facilitate our biobanking efforts. So why do we need a biobank for HHT? Right now, and historically over the years, we have animal models of disease. So there are mice that you can sort of give HHT to in a way. We also have cells that we can knock out copies of certain genes and watch how they grow in a dish, or we can simulate different ways to design a blood vessel in a dish. But those models don't perfectly replicate the way that HHT actually presents in a human. And researchers have told us over and over again that human samples are the thing that they need the most to answer the unknowns about this disease, to fully understand the mechanism of how arteriovenous malformations form, and to figure out which types of treatments might be best for the most people with this disease. Also, if you can imagine, you are a researcher, maybe in Idaho, and you want to run a project with HHT looking at how the AVMs in your lungs form and progress over time, and you need human tissue to study that. You can write a protocol, submit that to your university ethics board, and then you can collect any surgical tissue from patients who enroll in your study from your university. But to get a significantly meaningful amount of tissue to actually study and come to a conclusion about something, that could actually take years to be able to collect, because HHT is a rare disease, as we know, and people aren't getting surgeries that regularly to actually have a lot of samples come from one place. So having a central biorepository like the CureHHT Biobank kind of solves that problem, because we can reach out all over the country, collect samples from patients with HHT, bring them to one central place, and then distribute them to the researchers who need them. But what's even cooler is that we can split up the samples. So we can actually make your research donation go further. So if you give 5 tubes of blood during a research event or a blood draw, we can split those tubes into multiple different samples that we call aliquots. So maybe 5 tubes of blood get split into 12 or 15 different tubes, and we can have some to keep in the biobank and some to send out. So that means that your research ideas don't have to wait for years for tissue to be collected at one site. We can get things going a lot faster. And Cassidy is going to talk about the types of things that can be donated.
Thanks, Cassie. So, because HHT affects blood vessels everywhere in the body, there are a lot of ways to help. Some of the type of donations we're looking for include blood, saliva, skin telangiectasias, placentas, surgical tissue, transplanted organs, postmortem donations, and philanthropy and awareness. And in the next set of slides, I'll kind of go over the process of what it looks like to donate. So that first major step is to express your interest. Once we know you're interested in donating, we can then move on to the informed consenting process. So during that time, our main priority is to make sure you understand the purpose of your donation and answer any and all of the questions that you may have. It's also important to note that if you do change your mind, you can withdraw your consent at any time, even if you've already signed the consent form. So once the consent form has been signed, both parties will receive a copy of that signed consent form, and then we'll move on to the enrollment portion. So during that enrollment phase of the donation, we will collect a brief medical history that pertains to your type of donation, and we'll also collect some information from your doctor because we will be working closely with them throughout the donation process. And in the next set of slides, we'll go over the different types of donations. So if we're collecting surgical tissues, again, we are going to be working closely with your surgical team and the hospital's pathology department. If we can have a little bit of a notice, that's always great, just so we have some time to send the collection kit to the hospital. But once we have the surgical tissues collected, we'll work with the hospital staff to package up those samples, and then they will be overnighted to our lab. And then in the next type of collection, blood and saliva, again, that can look a little different depending on where you're having these donations done. So if we are having a patient conference or a pop-up event, we typically handle the packaging and gathering of the blood and saliva donation. And it's important to note that we can do these types of donations before a scheduled surgery. So once the samples have been collected and packaged appropriately, we'll send them back to our lab where they are centrifuged and separated into plasma from the blood cells, and then that plasma is then frozen and stored for future research. And then placenta donations, while is a little on the trickier side because babies make their own schedule, our main goal is to help make sure you have a safe and healthy delivery and a happy, healthy baby to give all those newborn snuggles with. And then on the back end, we will be working closely with your delivery team and the hospital staff to make sure the placenta has been collected and packaged appropriately. We typically take 6 samples across the placenta, plus anything that looks unusual. And then it's important to note that the placenta can be donated even if the mom or dad has HHT, because the placenta is an organ made from the baby. This type of donation is incredibly valuable right now because it helps support our women's health initiative studying HHT from reproductive years through menopause. And otherwise, the placenta would go to medical waste. So, again, incredibly important that we have some placenta donations. And then, as far as what happens to your samples, is the samples are typically stored and split into several smaller aliquots, like Cassie mentioned earlier. Your donation is coded with a subject ID and grouped with others for research. You will not be contacted unless medically necessary. However, you can opt in to hear about future research opportunities during that enrollment phase. And then the last type of donation we have is, again, that end of life or post-mortem donation. So CureHHT partners with Anatomy Gifts Registry, or AGR, which is a nonprofit organization supporting research and medical education. You can make a directed donation to HHT research, but beforehand you would need to complete the paperwork with AGR ahead of time, as well as let us know. And then also let your family members know what your wishes are as far as donations and end-of-life care go. And that's a brief rundown of what the process looks like and what type of donations we're looking for. So I'm going to send it back to Cassie.
Thank you. Awesome. So, a lot of different tissues that we can accept. HHT is a disease of the blood vessels, and we have blood vessels all over our bodies. So that's why there's such a great variability in the types of tissues that we can accept in the biobank. And this is a little bit about what we could use the tissue for. These are actually straight from the working groups of the CureHHT Research Network that developed our research roadmap. Every single one of those working groups stated that the biggest tool that they needed and lacked was human tissue in a central biobank, so that they could better understand the variability in HHT. What this means is that HHT is typically inherited every generation, but not every member in the same family with the same inherited mutation has the same symptoms. So your dad with HHT might have a brain AVM and a lot of nosebleeds, while you may not have any nosebleeds, but maybe you have liver vascular malformations, but your brother, who's also genetically positive for HHT, has no symptoms. That variability needs human tissues to study, and we'll talk about that a little bit more later. Also, like Cassidy mentioned, the Women's Health Initiative, uterine involvement in HHT, we need these tissues so that we can better understand what's happening with women with HHT who experience heavy menstrual bleeding, and how that changes their clinical management and the way that we care for those patients and follow them over time. Understanding brain AVMs, which ones are more prone to rupture and hemorrhage. There was a long-standing study called the Brain Vascular Malformation Consortia that was run over the course of 16 years that just ended this past year. And in that study, they were enrolling patients with HHT who had brain AVMs and those who didn't. And when people had surgery to remove those brain AVMs, they were collecting those. But the funding for that program has now gone away. They had a great 15 years of funding. And so there's no one actively collecting these tissues. So now we can take them into the biobank so that they continue that really important work to better understand brain AVMs. We can also use samples to identify gene variants. So we think we pretty much have a handle on the genes that cause HHT, but there are different variants in each of the genes that make up all the different types of mutations that people can inherit. And some of those, if you are a patient or you know someone who has received a genetic test result that is inconclusive, but clinically they've been diagnosed with HHT, sometimes we find those through studying biological samples. And then we can also look at the liver to better understand who with liver vascular malformations may go on to develop cardiac symptoms. That's a really important thing that we don't know yet, and we're trying to actively work on. We just launched our liver and heart working group. Same thing with pulmonary AVMs and their outcomes. And then we use samples to establish cell lines. So you can take samples from the blood and you can manipulate them in the lab to keep them alive long enough that you can treat them with different drugs or different compounds to see how they behave. And this is kind of the very first start of the drug discovery and drug development process. And it's a very important thing to do so that we can get more therapies into clinical trials for patients. So looking at the biobank and where we are now, we started the biobank in the summer of 2024, but we really didn't advertise it at all. We've just been passively collecting things from patients who have told us they'd like to donate or that they're having an upcoming surgery. We've also been collecting at our patient conferences where people gather and we'll bring in a phlebotomist to collect blood. So we have 119 individuals enrolled into the biobank. And from those 119 people, we have over 600 biospecimens, which is really great. And we've been able to distribute about half of those. So we wanted to be able to build up the amount of tissues and samples in the biobank, but we don't want to hoard them. We want to be able to give them away to research teams, but we have to make sure that it's a right fit, that the samples that we have are suitable for their research needs, and that we have enough of them to make it worthwhile for them to answer their question. So if someone has a proposal and they need 20 samples of something to be able to statistically say, you know, this is their hypothesis is proven or not, and we only have 10 of those samples, maybe we keep collecting and then provide those to them or something. So that's kind of how that process goes. The most things donated so far are blood, which we take the plasma, which is like the liquid portion of the blood, and freeze that. We have a lot of lung samples, a lot of liver from liver transplants, and then, like Cassidy said, placenta. And I did put this here at the bottom. You know, every sample started as someone's gift. And I know that sounds really cheesy, but it's really true. And it may seem like nothing to donate discarded medical waste or, you know, a couple tubes of blood to the biobank, but it's very meaningful to be able to donate to research, especially for a rare disease like HHT, because actual discoveries are being made. And it's kind of crazy to see that progress in just the last couple of years since starting the biobank. So I'm going to talk about some of the published works using samples that we have helped, you know, from people wanting to donate, get into research labs, and help make a difference. So the first and the second one, actually, both of these come from the Marchik lab, Doug Marchik from Duke University. So this goes back to that question of why HHT is so variable among members of the same family who have the same inherited genetic mutation. So everyone who has HHT is born with one broken copy of the gene. So you inherit two copies, one from your biological mom, one from your biological dad. And if your father has HHT, the chromosomes that you inherit from your father, every one of them has that broken copy. But if your mother doesn't have HHT, all of those copies are normal. So you can see the X for the inherited mutation, and then the blue chromosome is normal. So that's usually okay. Like I said, you have blood vessels all over your body, but you typically don't have blood vessel malformations all over your body. They're just sort of sporadic and variable in certain places. So it's generally been assumed that one functional copy is enough for your cells to do a good job at making your blood vessels. But something happens and you get a second hit. So you have this first inherited broken copy, and then something happens and you lose the second functioning copy of that gene. Maybe it's the gene endoglin if you have HHT type 1. So without that functioning endoglin, you can have an AVM form. And the second hit is what we call a somatic hit. That means that it's just localized, just in that cell. It could come from trauma, random mutation, inflammation. We get mutations all the time. That's why we have backup copies of things, but it's enough to cause an AVM to form. So to study this and to figure out if this second hit hypothesis is true or really plausible, you need human tissue, because you need to be able to look at tissue that has an AVM in it and tissue that has normal vasculature. And if this is true, the normal vasculature, every time you do genetic sequencing, you should just find one copy of a chromosome that has a germline mutation, that inherited one. And then when you look at the tissue that has the arteriovenous malformation, you will find both copies are gone. So the lab did just that. And you can see the space right here is a part in an endoglin gene that has both copies kind of down, or it's a loss of function in this part of the gene for both of the chromosomes. And then they ran it again a separate way, a separate time, and they found the same thing. So they discovered it here, and then they validated it in this one. And this first publication looked at tissues from patients who had HHT type 1 and HHT type 2. And we know that there are a couple other types of HHT. So the third type is the third most common, but it is definitely a lot less common than HHT type 1 and 2, and that's SMAD4. So those who have a mutation in SMAD4 also have another disease called juvenile polyposis. They receive tissue from a surgical resection of a very aggressive AVM in a SMAD4 patient, and they showed the same thing, that there was a second hit or a loss of function in both of the chromosomes so that an AVM was formed. So this is the first evidence that the second hit theory is actually, found in tissues. Those somatic mutations are present in the arteriovenous malformations. And it's important because it changes the way that we understand this disease and the way that we approach therapies that will be applicable and relevant for all types of HHT patients. So this was really cool work. And then speaking of therapies, the third one is about a vaccine. So this is a very cool paper. You can measure things in the blood, or you can measure things in the DNA. You can do single cell sequencing and look at expression of different kinds of proteins and different genes. And you can see things that are irregular in patients with HHT. One of those things is Ang2 or angiopoietin-2. It is typically a little bit higher in the blood vessels of people or animals affected with HHT. So this lab, Philippe Morimbaud's lab in New York at the Feinstein Institute, was studying if a vaccine against this angiopoietin could help reduce or prevent AVMs from forming. So this was a target that they saw was elevated in the vasculature of mice with HHT. They were able to get some biobank samples to confirm that Ang2 levels were actually different and higher in real people who have HHT compared to healthy controls. And then they created a vaccine against this to try to normalize that level in the mice. And so you can see here, this very top part right here shows you kind of the different types of mice. So it says PBS, it's like saline. And then over on the right, you have this BMP9. So this is basically telling you that they have injected the mice with something to make them have an HHT-like disease. So BMP9 is the HHT pathway. And then you have to have a control group. So they also gave a shot of saline to another group of mice. And in the group of mice that they gave the saline to, they looked at their vasculature after they were given saline or given the vaccine. And there's no difference. So it's showing that the vaccine isn't harmful to the vasculature of those mice. But in the mice that now have HHT, they either give saline, and you can see all the telangiectasias in their vasculature because they are HHT mice not receiving a treatment because it's saline, or they give them the vaccine, and you can see that those telangiectasias are gone. So this is really exciting data in a mouse model of HHT. And we need more confirmatory data, probably some more testing on human tissue or human cells in a dish. And maybe this can move forward into some type of trial. And then we've also been able to help some of our upcoming clinical trial groups or groups that have just launched their clinical trials prepare. So in a way that, the last lab that I showed you was measuring different biomarkers to see what was elevated, what could potentially be a target. Clinical trial groups need to do the same thing so that they know what to measure as an outcome in their clinical trial to make sure they can measure if their drug is working or not. So they need blood samples from healthy people, people with HHT, different types of HHT, so that they can better understand the baseline differences and what those biomarkers are. And then they have sort of a range to go by when measuring them in their clinical trial. So they can tell what would be sort of a meaningful change in those biomarkers. And it's also really important to have, lots of little samples of that blood to sort of calibrate things. So whenever you're developing these assays to measure certain things, you're going to have to sort of develop it based on, something that is true. So they take blood samples for those types of things, which has been really fun to help facilitate. So we've talked a lot about the types of tissues and what they're used for, but I haven't talked much about when you can donate tissues. I know a lot of times people go to the doctor and they donate blood. They don't donate blood, but they have to give blood for standard of care. They're getting their labs done. It's not so easy for us to just add on a tube of blood to those research or clinical blood draws right now, because biobanking is, it's not cheap all the time, especially shipping things back and forth. So it's typically easier to donate blood whenever we ask for volunteers, if we're having a pop-up blood donation event, or if we're having a patient conference and we have a group of people together. It makes it a lot easier financially for us to be able to facilitate a whole group of people and shipping those samples to the lab and doing that post-processing than one by one, very sporadically. So we have to make sure that we're good stewards of the money that's been donated to the foundation to allow us to start the biobank. And that's why we can't just, take blood samples added on every time someone goes into the clinic to get labs done. But we're working on ways to try to facilitate that in an easier way that's more logistically easier and easier on the finances. But definitely after any planned surgeries, or if you are pregnant, even if you are a woman who's had a hysterectomy in the last few years, they keep tissue samples in pathology departments at the hospital where you had that surgery for a certain number of years. And it's very likely that you still have samples at that hospital where they looked at it to determine, if the tissue looked okay, or to make a diagnosis of some sort. We can actually still pursue those tissues. If you're interested in having them transferred to the Cure HHT Biobank, we can help facilitate that. And then we can make nice microscope slides out of them for our Women's Health Initiative to study. And then, of course, like Cassidy mentioned, the body donation services, that Anatomy Gifts Registry helps to facilitate for us. A lot of times, if you donate your body to a university program, it's more of a general needs program. So you may go into whatever the biggest need is for the medical education community and research in general. They typically don't do what we call directed research, where they allow certain body parts or organs or tissues that are affected by a disease that you have to go to another location. It's always still good to donate your body to a university program or something like that, because even if you can't make a directed donation happen to study your pulmonary AVMs or something after you pass, if that's your wish, the medical students or the trainees who work on your body or to study your body, they will learn about HHT in the process, and I guarantee you they will absolutely never forget it. I will never forget anything about my human cadaver that I had whenever I was in grad school, and so it's a noble thing to do either way you go. If you want to donate to a body bequeathal service that does directed donation, or to your local university for medical education. Again, the more time that we have to prepare, the better. But Cassidy and I have gotten pretty good at this so far. So, like she said, sometimes those babies just don't wait, and we have been able to rally and pull things together within an hour or two, so we can make it happen. But I just wanted to express my very sincere thanks. Everyone who's donated tissues so far or told their family members about this, people who have donated money to get the biobank started, or people who have just encouraged us along the way, because it can be a challenging thing to figure out how to do and the logistics of it. So the hospital staff who help us all around the country, they deserve a huge thanks too, because they really do a lot of work alongside of us. And then, of course, the LIST lab at Boston, they have just been absolutely fantastic stewards of our tissues and helping us to manage everything. So if you all are interested, I'm going to leave this slide up for just a few minutes. So you can scan this QR code. You can fill out our research interest form. We can contact you with research opportunities, or you can let us know if you have a planned surgery or if you're interested in learning more about the biobank. You can always email us at research@curehht.org. That goes to both me and Cassidy, and our emails are down here. Of course, go to the main website if you ever want to stay in touch, look at the upcoming clinical trials or research studies that are ongoing. And then, of course, you can always give us a call. Phyllis loves to chat. I do too, but Phyllis takes a lot of the calls, and I'm sure most of you guys that are on here already know her. So thank you.
Thank you, Cassie and Cassidy. That was great. So we're going to head into our Q&A session, which everyone enjoys. And some of these questions you may have covered during your presentation, but we have people popping on and off. So it'd be great to just cover some of the general questions again. And so if you have additional questions, please feel free to put them in the Q&A, and we will try to get to as many as possible. Whichever questions we don't get to, we'll follow up with an email to try to answer those for you. So one of the questions that was asked by several people is about donating blood and blood samples, and is there a cost involved? So I know you touched on that a little bit. So is this just donating at your doctor's office? Explain a little bit more about how that works.
Yeah, I can take that question. So there is no cost to the patient to donate. As far as where to go, I think I might have mentioned in the slides earlier, we do occasionally have patient conferences and pop-up events where we do accept blood and saliva donations. So again, hopefully we'll see you at a patient conference or a pop-up event, and we'll be happy to help facilitate that donation there.
Great. And a couple patients were asking about tissue donations. Does that have to be done through an HHT center, or can it be done at a doctor's office or surgical center that's not an HHT center?
So one other thing I didn't mention earlier is we are currently working on some contracting with labs like Quest or LabCorp to hopefully be able to make this more accessible and open to the public to go to either a Quest or LabCorp lab, have your blood drawn there, and then we would work with them to have those samples sent back to our lab and they would be processed there. So we're in the
Great.
works. We'll let you, hopefully, we'll be able to make an announcement very soon when that's up and going.
Yes,
Now,
and
what about internationally? I mean, we know we have centers throughout the world, and how are we able to facilitate international donations to our biobank?
So, unfortunately, right now we don't have the means to do international donations. However, if you are in UK or wherever the case may be, and you know of a facility that does these type of donations, we'd be happy to help get you connected and facilitate that communication.
And our centers in Canada, the patient specifically asking about our Canadian HHT centers, are they able to help with getting donations or samples?
So I don't believe so in Canada either. Cassie, that's correct?
yeah, right now our biobank protocol is set up around the regulations and requirements in the United States, and they are different from country to country or nation states within the EU. And so, for simplicity, as we were starting the biobank, we wanted to start in the United States. However, Canada would be a next logical choice for us because the shipment of samples to the biobank from Canada would be a lot easier than, say, from Europe back to the United States. Just to give an idea, it costs $200 to $300 to overnight a biospecimen that's been collected. And so, if you're talking about international shipments, that price goes up exponentially. And so, we're, like Cassidy said, we like to help facilitate researchers who live in Canada or who live in the EU or other parts of the world. If there are patients in their area who are interested in donating research and their labs are able to accept human tissues to study, then we can kind of bridge that connection locally without going through the Cure HHT Biobank. So we're really more like a facilitator in that case than a collector.
That makes sense. And what about what other, we talked about blood samples, and what other types of tissue do you or do you ask for the biobank? What other types of tissue samples for people that are having procedures done?
Yeah, this is, it can be kind of a tricky question because, HHT does affect a lot of different places in the body. But again, it's one of those things where we do have to be a little bit specific in the types of tissues that we take because we want things to be collected in a way that's standard so that the research and the data that comes from it is nice and clean and usable. But also, we want to make sure that it's relevant for what our research community is studying. So, for example, if you're having a surgery or, like you're getting some moles removed, those are just normal moles or skin biopsies that are unrelated to HHT. That type of tissue is probably not going to be relevant for HHT research. If you are getting telangiectasias removed from your face or another part of your body, depending on how they remove them, those are very relevant types of tissues that we could collect. Basically, we want things that are either blood, some kind of fluids, or that have HHT affected vasculature within the tissue that we can study.
No, that makes sense.
Oh, And this can happen at an HHT center or anywhere else. So if you don't regularly go to an HHT center or you're having your procedure somewhere else, you can just let us know and we just work with your medical team. And we've had pretty good luck so far. I would say most of our tissue samples come from non-HHT centers.
So how far in advance? Let's talk about a patient who is pregnant and they want to donate a placenta. How far in advance do they need to contact you about that?
So this, I would say the sooner the better, just so we can make sure collection kits get to the hospital in time. But again, like we've said, babies make their own schedule, and anything can happen. So we've gotten pretty good at getting things done within a 2 hour time frame. So we try not to do that, but we do what we can.
Yes.
Sure. And what about so another question we had from a couple of patients is do families find out like if they donate the samples or tissue or blood, do they find out if it was used and what it was used for?
Yeah, that is, we typically don't go back to families and say, hey, your tissues were just sent to this research team, or this research team just published this work and it included your tissues, because your privacy is very, very important. And, giving away details like that is typically just not appropriate for making sure that donors stay anonymous. And we also have meet all those regulatory requirements that we're bound to. We do like to let you know when your samples have arrived safely at the biobank so that it's kind of not a worry, and they're not in the mail somewhere or in transport somewhere. But beyond that, you can look for general updates on publications from the Cure HHT team, and we like to highlight these on our website or in blog posts. And it is a requirement, any tissues that come from the Cure HHT biobank that go to research teams, we know what the research is about and what they're using the tissues for. And they also are required to let us know when they publish any research so that we can summarize that for the community to let you know that your donations are being put to good use and that there's actual fruit coming from all of this work.
Are there some current projects that that you can share with us that donations have gone to that we're getting ready to launch or announce or people are working on?
Yes, there are quite a few. I can't share them yet because I can't steal their thunder, but there are some very exciting projects for, just basic mechanistic research all the way to clinical trial research and further drug development. And we're working with a couple of new groups who are thinking about joining the HHT space in the industry and pharma world, but they need some patient samples to test and just make sure that it would be feasible for the drugs that they currently have to be tested on patients with HHT. So that's kind of the first step there. But those groups aren't openly studying HHT at this time. So we will let them evaluate the prospects and then come to the table whenever they're ready. But I will say that there are a lot of abstracts at the upcoming scientific conference in October that use human tissues, and we will publish an abstract summary in Angiogenesis, and we will also recap it for our HHT community, and we can tell you then.
Oh, that's great.
Mhm.
What about now? We have a lot of researchers and scientists on the Zoom today, and some of them are asking, so how do they go about obtaining samples?
Ooh, yes. So that is a great question. First, I would love to hear from you. You can email me directly. I need to also refresh my survey. Whenever we first started the biobank, I had reached out to a lot of our researchers in our Cure HHT research network to ask who needs human tissues to study? What types of tissue do you need? Do you need them to be prepared or processed in a certain way or collected in a certain way? We want to survey the community like this so that we can ensure that our protocols in collecting tissue meet the widest range of need in the research world, because we want to be as applicable to as many people as possible. So we've had quite a few new researchers join the HHT cause, and so it's on my list of things to do in the coming months to send out that survey again so that we can continue to refine our collection protocols. And then whenever we have tissues that meet the needs of your research aims, we can facilitate the transfer of those tissues to your team so that you can study them. Also, if there's something very specific that you're looking for that is kind of outside of the biobanking area per se, so say you want to find 50 people with HHT and you need half of them to have an endoglin mutation and have pulmonary AVMs and a brain AVM. Then we might need to collaborate, create a different study, and then we can seek out people with those eligibility criteria and try to collect samples from them instead of waiting for those samples to come to the biobank. Because the biobank is really about collecting tissues to later distribute. It's not about recruiting a certain type of tissue. So it's a very clear line there. And we sometimes like to collaborate and develop other studies so we can go after more specific types of tissue in a prospective way, if that makes sense.
So we were talking earlier about patients and the de-identification within the biobank. So talk a little bit about our privacy standards and our data collection and how we keep that confidential.
Yeah, so like previously stated, each participant is given a study ID, so your name and information is not used for anything. That ID takes place of your name. Before a study is up and running or enrolling, we have to go through an ethics committee or the IRB. So they review all of the documents associated with the study. So that could be our informed consent, the protocol, things of that nature. And those are all reviewed and approved by them. And then throughout the duration of the study, we have, I guess you could say, check-ins where they review the data that we've already collected and other documents. So,
That's good to know.
yeah.
So I'm going to ask a little bit more of a specific question that a patient proposed. So they asked, migraine is one of the most common symptoms of HHT according to the CHORUS study. And what can the biobank study tell us about this?
I love this question. This is such a good question because you're right. The CHORUS study, which is our comprehensive HHT outcomes registry of the United States, it is a clinical registry that is in 16 of our HHT centers, and it basically just captures everything from head to toe related to HHT and the symptoms that you have, any kind of treatments, imaging, lab values, you name it. And from CHORUS, we know that people with HHT, majority of them, I think it's like 56%, suffer from migraines. And most of those people who have migraines also have pulmonary arteriovenous malformations, but not all of them. Some of them are also anemic and have pulmonary arteriovenous malformations. Some of them are just anemic. Some of them just have migraines without any other symptoms. And so the registry basically tells us what is happening, but the biobank can help researchers begin to ask why something's happening. So to do that, though, we need a researcher to come along and say, I have this research proposal. I want to better understand migraines and HHT. Are there certain characteristics, maybe blood biomarkers, inflammatory cytokines, something weird with the vascular structure that's causing these migraines, what are the things that I can look at in the tissues from the biobank to sort of put together the why behind the migraines. And then once you know why, you can maybe develop treatments or preventative methods for that.
So that's how a lot of this moves forward,
Yes.
with all of these different manifestations. How about the cost of the biobank? I know we said it's not a cost to the patients, but how is that funded?
Right. So science is not cheap, and that's unfortunate, but it is true. And we've had some very amazingly generous donors step up and fund the biobank. And that includes contracting with the LIS lab, kind of renting their space and their excellent smart brains and expertise, and then also the shipping cost. And then if we hire a phlebotomist to come to our patient conferences, that's it's a lot of different things that are combined, and it's all patient donations that has made this biobank get started and happen, which is absolutely incredible.
Yeah, so everyone sees how their donations are actually at work, which
Yep.
is really terrific. Let's see. I don't know if we had some other questions. So someone was asking, they're having an aortic resection. Is that a type of tissue that might be feasible for the biobank?
Yes, I would take that.
That's good
Yes.
to know.
If you are interested in donating that tissue, that is something, because it is vascular related. We would be interested in having that for the biobank and making sure that it got into the hands of a researcher that could, use it in their research in a meaningful way.
All right. Well, Cassie, you want to put up your slide again about how people can get in touch with a QR code and all of that for people that popped on late, so everybody has a chance to see that. This webinar is being recorded, and you will receive a link to be able to watch and go over any of the slides again that Cassie presented.
Okay, just a second. I have too many monitors and I have to situate everything so I can find it all. And we can also, we will send out the link to this survey, the research interest form. We'll send that out to everyone who's registered, and it will also be on our website. And again, you can always email research@curehht.org or myself and Cassidy, and we can help answer any of your questions. If you're a researcher and you're looking for tissues, let me know. I saw some good questions in the Q&A box about that, and we'll get back to you with more specifics on those things. And then also remember, if you are a healthcare provider and you have a patient with HHT, who's interested in donating tissues, just, give them our contact information, or you can ask their permission to share their contact information with us so that they can ask questions and learn a little bit more about it.
That's great. And for those of you on the webinar today who are your family ambassador, please share this information with other family members of HHT, so they can reach out to Cassie and Cassidy about making that really important donation. I want to thank everybody for attending today. Please send any questions that didn't get answered. If you have anything else that you think about later, please email Cassie or Cassidy, and they'll be happy to get back to you. From all of us here at Cure HHT, we appreciate your interest, and everyone that has donated, whether it's tissue, blood, whatever it might be. We are so grateful, and we hope you'll join us for our next webinar. Thank you so much. Have a good night.